Yeah, I'd just like to start by saying thank you to Chris and to the organisers for having me and Laura come over to the LMB to speak to you today. It's really good to be here. Myself and Laura are both research scientists based at AstraZeneca, so the building across the road. And we're here today to just talk around thermal shift methods with a particular focus on those methods that we apply within our department, within high throughput screening, because they can be employed at scale for the purposes of screening in early drug discovery. So I thought I'd just start with giving a bit of background around our department and what we do. So what is high throughput screening? So high throughput screening, we sit within the wider department of hit discovery at AstraZeneca, and we comprise a very early stage of the process that goes into discovering a potential new drug. So within our department, once a target has been identified and validated as having a particular role within a particular disease process, we're responsible for assay scale-up and rapid screening of large numbers of compounds against these targets, which come from AstraZeneca's internal compound libraries. And this is to identify compounds or chemical series which are active against those drug targets. And by active, we mean anything that produces kind of a measurable response in that target. So it can be either modulating its activity, so inhibiting that target, or it could just be binding to the target. And once we've identified these active compounds, those are then progressed to later stages of the drug discovery process to be further kind of chemically optimized and confirmed, and hopefully become start points for potential new drugs. So a good analogy is that HGS functions kind of like a large funnel. So we carry out very rapid screening of hundreds of thousands of compounds at single concentration, and then actives from that screen are then taken forward to further rounds of confirmation. And as we kind of progress through, use my pointer, progress through the HGS cascade, we kind of narrow down these numbers until we end up with tens to hundreds of compounds comprising up to 4 chemical series, which are then progressed to later stages of the process. So to carry out this large, very rapid screening in a reproducible manner, we rely a lot on robotics and automation. And if you walk past kind of the ground floor of our building over the way, you should be able to look in and see these robots, hopefully with green lights on, running in the lab, running our assays for us. So what kinds of assays do we run within HGS AstraZeneca? The kind of assays that we run have really evolved directly based on the targets that we've been required to screen. So quite historically within AstraZeneca's portfolio, we've had a lot of catalytically active targets with well-defined binding sites. So this has allowed us to be able to use assays which monitor functional activity, for example, inhibition of ATPase activity, or because we know, where the binding site is, we can design probe-based assays to interrogate binding to that particular site on the target. However, as these graphs are kind of showing, towards more recent years, we've seen a shift in the portfolio towards what we'd class from a drug discovery perspective as more challenging targets. So that's quite a broad definition, but it generally encompasses targets that are hard to prosecute by traditional screening methodologies and assays. So these targets might lack any structural information. They might have no functional activity, so be catalytically inert, which means we can't kind of monitor that in an assay. They might be disordered, contain disordered domains, or they might lack any sort of known chemistry or known information about binders. And usually we'd use tool compounds to kind of validate and build our assays. So this has been quite a challenge. And in order to kind of tackle these increase in challenging targets, our shift in methods have had to adapt in response. And a large part of tackling these new targets has been through running biophysical methods, which monitor affinity-based methods, which monitor binding of molecules to your target. So for these methods, you don't need any sort of functional activity. You don't necessarily need tool compounds to build your assay. And you don't really need structural information because they're unbiased. You can look at binders at all possible sites on your target. And the 2 main biophysical approaches that we employ within HGS are ASMS, which is an affinity-based mass spec assay, and we also employ DSF and CETSA, which are both high throughput thermal shift assays, which is what we're going to focus on for the rest of the talk. So before we properly get into things, I thought I'd just give a recap on kind of the principle of thermal shift assays and how they work. So thermal shift assays detect changes in the conformational status of a protein target brought about by heating. So this is just a very simplified representation of the unfolding equilibrium that exists between targets and their native folding state versus their unfolded state. And thermal shift methods are crucially able to discriminate between these two states of your protein. And yeah, they're able to monitor this unfolding equilibrium and how it shifts in response to temperature. And they can be really useful for finding out information about ligand binding. So if we add kind of the ligand binding equilibrium onto this model, for the most part, ligands will bind to the native folded state of your target to form kind of a protein ligand complex. And in doing so, it stabilizes this folded state of the target and shifts the unfolding equilibrium in this direction. So through monitoring this unfolding equilibrium and how it changes in response to different ligands, thermal shift methods are able to indirectly derive information about ligand binding through monitoring this. So this slide's just showing a bit of a timeline of different thermal shift methods and kind of when they were introduced for drug discovery. So the principles behind protein unfolding have been studied since sort of the late 19th century, but it wasn't until more recently that the role that heat played in that unfolding transition was kind of discovered. And in the 1960s, there was the introduction of the first differential scanning calorimeter. And this really allowed kind of thermal unfolding measurements of proteins to be undertaken more widely. I'm not going to focus too much on this because Chris covered this in the previous lecture, but just as a reminder, DSC kind of monitors the heat capacity of different protein states, and it monitors the excess heat, which is associated with conformational changes in your protein target brought about by heating. So this was really fundamental in allowing thermodynamic measurements of protein unfolding to be undertaken. However, it requires quite a lot of sample, quite high volumes, and this limited its sort of use for screening purposes. But the situation changed more recently. So in 2001, there was the introduction of a fluorescence-based thermal shift assay, which was applicable to screening in 384-well multi-titer plates. So yeah, because it's fluorescence-based, you can screen in multi-well plates, and it requires much less volumes of sample, per sample, much less protein consumption and therefore cost. So this really made the thermal shift assay applicable to high throughput screening. And this is an assay that we apply ourselves within HTS AstraZeneca. So what is dye-based DSF and how does it work? So like DSC, it measures the thermal stability of isolated protein targets. However, rather than looking at and monitoring heat capacity, it monitors fluorescence emission of extrinsic dyes which are present within your sample. And we typically use hydrophobic dyes such as Cipro Orange for this purpose. And how it works is that when your protein is in its native folded state, the dye which is present in your sample is exposed to the aqueous environment and fluorescent signal is quenched. However, when you start to ramp up along a temperature gradient, this begins to unfold your protein, and hydrophobic regions within kind of the core of your protein are exposed, and they're able to interact with the dye molecules non-covalently, increase their quantum yield, and you see this increase in fluorescent signal. And that's up until a point where your protein completely unfolds and aggregates, and dye fluorescence is quenched, so your signal drops again. So this phenomenon typically results in kind of a sigmoidal fluorescence profile, also known as a thermal melt curve. And we define the stage at which half the population is unfolded. That's known as the TM, or the melting temperature. And what's good about following and monitoring the TM is that it's sensitive to the conditions to which a protein is in, and monitoring how this thermal melt, the TM shifts, or the delta TM. This provides a measurement of the thermal stabilization of your protein under different conditions. So some of the main advantages of dye-based DSF are that it's convenient, sensitive, and it can be applied in a high throughput manner. It can be used for protein QC, just to check that your protein is properly folded. Also for buffer screening, to investigate if there are particular buffer components which confer stability. and this can be useful for long-term protein storage and crystallography. However, as we sort of discussed earlier, ligand binding is able, for the most part, ligands bind to kind of the native folded form of your protein, and they're associated with an increase in thermal stability, which manifests as a higher TM value. So through monitoring the shift in TM, you can get information about whether a ligand is thermally stabilizing your protein and infer whether it might be binding. And this is the way we apply it within HDS. So we use it to assess ligand binding in a small molecule screening setting. So I just thought it'd be useful to talk through some kind of considerations that you might want to think about if you were developing your own dye-based DSF assay, and share some of our learnings around this as we've been through the process. So some things that you might want to consider are the compounds that you're screening and the plate types that you're using. So within AZ, we screen an 100K compound set known as our DSF collection. And this is our standard screening set that we prosecute against different targets when we're running a DSF assay. This set was obviously kind of selected to maximize the structural diversity and compounds that we have in there because we use it for hit finding. But we also limited inclusion of certain types of compounds. One of those sort of classes is compounds with poor solubility, because we know that these result in a reduced response in the assay. And we also know there are some compounds which interfere with the assay. So compounds which are quite lipophilic or maybe intrinsically fluorescent can cause assay interference. So this is sometimes something that you might want to be mindful of if you're running your own assay. In terms of plate types, what's really good is you can use a variety of different PCR plates. They're all commercially available. We use 384 well plates, but you can use 96 well plates. We found that white plates are beneficial for signal compared to clear plates. And if anyone's interested, these are the two plate types that we've kind of settled on within HDS, and these are the ones we use regularly, and they perform well, give us good results in our assays. Another really important consideration is the fluorescent dye that you choose to use within your assay. So we use Cypro Orange, but there are many commercially available extrinsic dyes, and this is quite a nice table which summarizes some of the more common dyes that are used for dye-based DSF. Cypro Orange is widely favored, particularly for screening applications, because it has a high signal to background in the presence of native folded protein. It also has quite a high emission maxima. So it emits at wavelengths between 500 and 610 nanometers. And this is good because if you have compounds which are intrinsically fluorescent, they tend to fluoresce at lower wavelengths. So having this high emission maxima kind of prevents too much assay interference from the compounds that you're screening. One kind of major disadvantage of Cypro Orange is that it's not detergent compatible. So if you have hydrophobic buffer components or detergents within your buffer, this might not be the dye to use. But there are other dyes. So there's the molecular rotor dye DCVJ and this commercially available dye Proteostat. These have both been demonstrated in the literature to have been run in cases where you have high detergent. So these might be dyes to consider in that case. We've also mainly focused on monitoring thermal stability of protein targets, but you can also use dye-based DSF to look at stability of things like RNA. And in those cases, there's a couple of dyes. So there's Cyber Green, which is an intercalated nucleic acid dye, or Quantit Ribo Green, which is a single strand specific RNA stain. So you could use those in instances where you wanted to monitor RNA stability or even ligand binding to RNA. Sorry, fast forward. So in terms of protein requirements, we typically use between 1 to 5 micromolar of our target. As we mentioned before, the protein requirements for this assay are much lower than they are for DSC. So we use about 5 microliters of sample per well. However, when we're running 100K compounds, this still requires quite a lot of protein compared to kind of conventional HDS assays. So between 50 and 150 megs per screen. So yeah, this is still quite high, and this is the main reason why we screen a 100K set and not a larger set as we do for some of our other assay technologies. And finally, in terms of detection, so how do you read your plates? So great news is you can use any commercially available real-time PCR instrument. We use the QuantStudio 7 Pros from Thermo Fisher. But yeah, you just need any instrument that's capable of monitoring fluorescent signal in real time as you ramp up along a temperature gradient. We use a temperature range of 25 to 90 degrees. Most proteins are completely unfolded at 90 degrees, and this takes us around 1 hour to complete that cycle with a ramp rate of 1 degree C per minute, which is a fairly typical ramp rate for a DSF experiment. So I just wanted to talk a little bit about how we actually carry out the analysis to dive into DSF. Specifically, how do we calculate this TM value? So just as a reminder, the TM is defined at the temperature at which half the population is unfolded. And this is the parameter that we monitor when we're looking at uncovering whether our target has been thermally stabilized. So there are a variety of different methods to calculate the TM, some more robust than others. A really common approach, and one that we use ourselves in-house, is calculating the smooth first derivative of the melting curve signal. So this is plotting the increase in fluorescence, or the change in fluorescence, sorry, with respect to temperature. And your TM in this case can be identified as the inflection point in the melt curve. So, or the point at which you have the steepest increase in fluorescence signal with temperature. The main reason we use it ourselves is because it's quite robust. It results in quite a reproducible TM value, which is important when we're determining our assay statistics and setting activity cuts in our assays. It's quite a simple method, so you don't need to fit a curve. And it can be quite universally applied to a variety of different thermal melt transition shapes, which can be really useful if you wanted to visualize if your transition shape has multiple transitions. So sometimes in your thermal melt profile, as I've just mentioned, you can kind of see that your target has two unfolding transitions within, or two or more unfolding transitions within its thermal melt profile. And this is just two examples of targets that we've recently screened. When you're just looking at raw fluorescent signal with respect to temperature, this can be quite hard to visualize. However, when you plot the first derivative, you can clearly see two inflection points in the curve, which correspond to the two TMs for your two different transitions. Sometimes when you have multiple domains in your protein, which unfold at different rates, that can manifest as two transitions where you have two TM values. And if you add ligands into the mix, so there's these colored traces here, it could be really interesting to see what these ligands do and whether they preferentially stabilize one TM corresponding to one of the transitions. And if you know what these TMs correspond to in terms of your protein domains, this can give you information about where the ligand is binding on your target and where it might be stabilizing. So this is quite an interesting application of DSF if you have that information to hand. We utilize GeneData's screening software package to carry out these calculations and to calculate the first derivative for us. But you can calculate the first derivative of a curve in GraphPad Prism. And there are also a variety of open source analysis tools that you can use, for example, DSF World or Simple DSF Viewer. And you can also use these to fit more complex models to your thermal melt profile. For example, if you have a kind of a classical sigmoidal melt curve, you can fit a Boltzmann equation to that curve to generate TM value. So I just wanted to finish talking about dye-based DSF by showing you how we actually run the assay day to day when we're screening, and also giving some example screening data for you to have a look at. So it's quite a simple assay to run in comparison to other assays. We start off with our 384 well multi-titer plates, and these already contain our compounds, which are plated as singles per well. We then dispense our fluorescent dye on top of that, and mention that we use separate orange, and we dispense it using an Echo 555 automated liquid handler. So you might hear a little bit more about this in the next biophysics lecture. We then add our protein, which has been prepared in assay buffer. We spin the plate to make sure everything is at the bottom of the well. Add a clear plate seal just to make sure we're not disrupting any fluorescent signal that we're generating in the assay, and incubate for at least 30 minutes to kind of allow our ligands to bind and to sort of make sure they establish equilibrium. And then finally, we heat the plates and read using the real-time PCR machine. Typically, when we're screening, it takes us around 2 to 3 weeks to complete a screen of 100k compounds, and this is run at single point, single concentration. We then take our actives from this and plate those up into fresh plates, and then perform a confirmation round where we want to confirm that we're seeing those TM shifts on another occasion. And at this stage, we run a 5-point dose response to see if we have concentration dependence in the TM shift. And then finally, when we identify actives from this, these are passed on to a dedicated biophysics team, which kind of screen these compounds in orthogonal biophysical assays, most commonly SPR, but also NMR and LCMS as well. So when we're screening, our kind of capacity or our throughput is to screen 33 at 4-well plates per day. So this is around 12,000 compounds, and that takes us around 10 hours using 3 QS7 instruments. So this is just some example single concentration screening data for one of our targets that we call kinase X. You can see that we've run 11,000 compounds in this run, and this is the well or the sample number along the x-axis, and then this on the y is our TM calculated using the derivative method that we mentioned earlier. And we've set an activity cut for this assay, and this is based on what we think is a significant shift in TM, taking into account sort of the variation in the assay, looking at control wells containing no ligand. So you can see the majority of our compounds are sat around a TM of 45 degrees, and this is the TM of the protein in the absence of ligand. So the majority of compounds are having no effect, which is kind of what we'd expect for a screen and run at this stage of sort of the drug discovery process. However, you can see that some compounds are having quite, there are a lot of compounds above this activity cut. Some compounds are having really large effects on the TM. And while sort of the data analysis for DSF can be automated, we could just take all of these through. It's often useful to kind of visually inspect the melt curves of compounds, particularly those which are producing very large shifts in TM. So this compound, for example, is producing a shift of around 30 degrees. However, when you look at the raw profile, fluorescence intensity versus temperature, you can see that in the absence of compound, we have a really nice sigmoidal curve shape. However, when our ligand's added, we have this really high starting fluorescence intensity. And this is kind of indicative of our compound being artifactual or having interfering in the assay in some way. So it could be maybe binding to the dye and causing this high starting fluorescence. So we would typically flag this compound as an artifact. It's not really suitable for screening and determining an accurate TM value in this assay. So we would flag this and probably exclude it from further follow-up. We also see compounds which induce negative shifts in the TM. So a negative shift in the TM compared to the melt curve in the absence of ligand. One reason for this could be that the ligand is binding to a less stable form of the protein or the unfolded state. We tend to see quite a lot of compounds producing this effect in the assay, which kind of limits our capacity to follow them up further. But in any case, we're most interested in proteins that we know bind and stabilize the folded state of the protein. So we would tend to flag them and deprioritize these sorts of compounds in favor of compounds which are producing a positive effect. And this is an example of one such compound which is producing a positive effect, a typical active compound. And you can see that it's producing a TM shift of around 5 degrees, which is above our activity cut or threshold for the assay. And you can see the melt curve shape is pretty much identical to the shape of the curve in the absence of ligand, apart from obviously this shift in the TM. So this is the sort of compound that we would take forward to later stages of confirmation. So yeah, when we run confirmation, we take the actives from the primary, and what we're really doing is looking to confirm that we see a thermal shift again on another occasion, and also look for concentration dependence. So as we increase the concentration of compound, are we seeing an increase in the thermal shift? And this just gives us a nice feeling that the compound is binding or stabilizing our target. An important thing to note at this stage is that the extent of the TM shift isn't proportional to affinity in a DSF assay. You can have ligands with equal KD, which result in different TM shifts. And in sort of an equal manner, you can have compounds which have the same TM shift, which have different affinities. And it all depends on how your compound is binding and interacting with your target. So it's been noted in the literature that compounds binding with mainly enthalpic contributions result in smaller TM shifts than those binding mainly via entropic contributions. So this is important to note, and we tend to kind of leave any affinity determination. We use confirmation round as essentially, confirmation that we're seeing a TM shift on another occasion, but we tend to leave any affinity determination to downstream biophysical assays such as SPR. So obviously dye-based DSF uses dye, and when you add anything additional into an assay system, it has the potential to interact and affect that assay system. So it's important to note that the inclusion of extrinsic dye in dye-based DSF does add a second binding equilibrium to the analysis and can cause slight destabilization of your target. So I've just added dye binding equilibrium to the model that we were discussing earlier. So the dye obviously is a hydrophobic dye. It binds the hydrophobic regions of your protein, so it preferentially binds to the unfolded state of your protein target. So in doing so, if you add high concentrations of dye, it has the potential to pull the unfolding equilibrium away from the native folded state and towards the unfolded state. And you can see that in the experimental data that we've generated here. So as we increase the dye concentration up to 15 times, you can see that the signal increases because we have more dye present in the assay, but the TM actually shifts in a negative direction up to -4 degrees, so quite a significant shift. And because we're looking for binders which bind to and stabilize the native form of the protein, we want to kind of limit this as much as possible, and we tend to run as low a concentration of dye as we can get away with in terms of for signal. So we tend to run it between 2.5 and 5 times dye concentration, but this is just something to kind of be aware of if you're running a dye-based assay. So a more recent addition to kind of the thermal shift assay toolbox is label-free DSF or nano DSF. And this method actually alleviates the need to include an extrinsic dye within your sample. The throughput of this assay isn't quite there for us to employ it in a screening sense, but we do use it within other departments at AstraZeneca for things like protein QC and stability screening for different buffers and things like that. So I thought I'd just touch on it briefly. So label-free DSF, rather than monitoring the fluorescence of an extrinsic dye, it monitors the intrinsic fluorescence of tryptophan or tyrosine residues present in your protein structure. And what happens is, upon heating, protein unfolded changes the polar environment around these residues. So most commonly, you have internal tryptophan residues, which are present in a hydrophobic environment when your protein is fully folded. And as your protein unfolds, they're then exposed to the hydrophilic solvent environment, and this changes their fluorescence properties. And it usually causes a red shift in fluorescence or a shift in the emission towards a higher wavelength. And through monitoring the ratio of emission at 2 wavelengths, so they monitor the wavelengths 330 and 350 nanometers, this allows you to kind of measure how the wavelength shifts with an increase in temperature, and allows you to plot a thermal melt curve from which you can derive your melting temperature for that particular target. So some of the advantages of label-free DSF, we've mentioned it's a more direct approach than dye-based DSF, so it avoids the need to include extrinsic dyes within your sample and introduce that second binding equilibrium into your analysis. We use it for protein QC, for buffer screening. It can be used to assess relative stabilities of different protein batches or mutants. And we can use it as well, kind of on a smaller scale, to look at ligand binding in lower throughput experiments. A good advantage is that it's applicable to a wide range of samples, including those containing detergents that you can't screen in kind of a Cipro orange-based assay. So this has been applied to study like thermal unfolding of things like membrane proteins. Some of the disadvantages are that it requires a target molecule to contain tryptophan or tyrosine in order for you to have enough kind of sensitivity to generate a thermal melt profile. And dependent on the number of these residues in your protein target, it can be a lot less sensitive than diet-based DSF. It's not appropriate for ligands that fluoresce in the same region as your tryptophan fluoresces. And because this is kind of a lower wavelength than we use for a CIFAR orange assay, there's kind of a higher propensity for your compounds that you're screening to interact in this region and for their emissions to overlap in this region. So that's something to be aware of as well. Currently, the throughput, as I mentioned, of label-free DSF is quite limited. You do require specialist equipment, which allows you to monitor these two wavelengths with respect to temperature. The kind of most widely applied instrument is the Prometheus by Nanotemper Technologies, and this has the capacity of screening up to 48 samples in parallel. So still quite a high throughput instrument for the majority of cases, but from a screening sense, it's not quite there yet. I did go to a webinar a couple of months ago from a company called Protein Stable that have just introduced a label-free DSF method that utilizes 384 well plates. So we haven't sort of looked into this or brought it in-house or anything like that, but this could be an exciting new development, kind of increasing the accessibility of label-free DSF from a screening perspective. So moving towards the end of our thermal shift assay timeline, a really exciting new technology has been introduced recently that allows you to monitor the thermal shift of different protein targets in a cellular context. So I'm just going to pass over to Laura, who's going to talk a little bit more about that.
Hello, hi, I'm Laura. I'm a cell scientist, so that's why I'm going to cover the last little chunk. We've been running these cell-based thermal shift assays for a few years at AstraZeneca now, and they, just like the protein-based thermal shift assays, really help us to prosecute these really intractable targets that otherwise we couldn't prosecute. Okay, so cellular thermal shift assay, what is it? It was invented by a company that spun out from the Karolinska Institute. They're called Pelago Bioscience. And the original paper here is by Martinez Molina et al., if you're interested. They hold a license for the technology. So AstraZeneca at the moment have an agreement where we can use their technology because we pay a license fee. I believe if you're in academia, you don't have to pay that license fee, but this is going online. I would check with Pelago. It works in a similar way to protein-based thermal shift assay, except that you don't just have one protein in this assay. You have your target of interest in a cell, which is essentially a bag of proteins, and you've got to fish out your protein of interest. So here on the y-axis, we have to find a way to quantify the relative amount of folded protein in the experiment. And one of the ways, there's many ways that you can quantify your protein, your folded protein. There's lots of antibody-based detection methods and mass spec methods. That's what Pelago have the license the patent to license. So Western blot on the left-hand side, really simplistic, really low throughput. You just need an antibody against your target and you're good. There are some methods like single cell turf and the high content imaging that take advantage of the fact that actually you can start to harvest some phenotypic information from your cells at the same time as you challenge with your ligand. There are some methods here, reverse phase protein array and alpha screen, that are more suitable to high throughput because you can array lots and lots of samples on a glass slide, or in the case of alpha screen, you can challenge with one compound in every well of, say, a 384 well plate. Previously in HDS, we've used the alpha screen technology. It's developed by Revity. They've renamed themselves, they were PerkinElmer. You need 2 antibodies against your target, and you need them to be conjugated to PerkinElmer's donor acceptor pairs. So this works really well if you've got antibodies against your target. If you don't have antibodies against your target, which is entirely possible if it's a novel target that no one's really done any research on before, then you need a different technology. More recently, we've been using this nanoluciferase based method, which uses a high bit tag. So we add the 11 amino acid high bit tag onto the end of our target, and we express our target with that high bit tag. We then lyse the cells after we've done the thermal shock. We come in with large bit, and the luciferin. High bit and large bit bind with high affinity to reformulate the nanoluciferase. We add luciferin and you get a luminescent signal. And the more luminescence you see, the more folded target you have at the end of your experiment. There is a box up there at the top right that talks a bit about mass spec. Mass spec is high throughput in the sense that you can look at lots and lots of different targets in your cell. So your global proteome and how it's affected or shifted by challenge with, say, one ligand. But in high throughput screening, we want to screen lots and lots of compounds against just one target. So it's useful further down the drug cascade. So all of the data that I'm going to talk about in the coming slides is based on the high bet tagging method. Is what we've been using more recently. There are different formats to a thermal shift assay, a cell-based thermal shift assay, and different variables that you can change in your experiment. So on the left-hand side here, we've got the sort of classic melt curve where actually you can change the temperature, and at a range of different temperatures, you can generate this melt curve. Unlike the protein-based DSF assay that Poppy was talking about, which is a continuous assay where you heat and heat and heat your protein and it melts. Actually, because we have to come in with detection reagents, we can only run this as an endpoint assay. So even though that is a continuous curve, we run this as a discrete set of experiments with every temperature in a different well. And that's a massive faff. We wouldn't want to do that for screening 500,000 compounds, which is what we want to do. So we have to adapt the assay to run in high throughput. And I'll come on to that in a later slide. Another way that you can run the assay is to pick a single temperature and test a range of concentrations of your compound of interest. And in doing so, you can find a concentration at which your compound is able to stabilize 50% of your target. If you do that, you have a measure of the potency of your compound binding to your target. And you can use that to rank your compounds of interest when you get towards the end of your screen. And you want to know which your most potent compounds are. Because this is in a cell, there's a third dimension to this experiment, which is the cell itself. And so you can perturb those cells any way you like. And so people in the literature, there's lots of papers where people have done different cool things like push cells into particular stages of the cell cycle or different polarization or differentiation states. You could run this experiment on a knockout cell line or a wild type cell line, a lysed cell. If you run an lysed cell, then you overcome issues with membrane permeability of your compounds. If you run in a whole cell, then you've got an intact native environment that's physiological in theory. So this is a real advantage of the cell-based thermal shift assays. How do we run these in high throughput screening? I just said we can't run the experiment at every single temperature. We'll be here for months. So instead, we pick a single temperature to run the experiment at. And the temperature that we're aiming for is a temperature where the signal is quite low, that would come from the native folded target because most of that target has been melted away. And there is a potential to measure any stabilization that you might see resulting from compound binding. And so we like to define something that we call the assay window, which is a theoretical min and theoretical max of your experiment. And on the right-hand side here, you can see that the purple triangles, these are examples of wells that just contain native folded target and the signal that you would get from that. And here are some wells that contain some control compound that you know to bind to your target of interest. And that defines the space that you've got to detect potential stabilization from all of these different black compounds that we've challenged with. In reality, we're working on novel targets and we're trying to find binders to targets that have often got no known chemistry, no known engagers of that target. And so the reality is we don't have that maximum to build a sense of where our assay window sits. And we've had to learn ways to develop the assays to account for the fact that we don't have that, but we need to still know that our assay is robust enough to run. So here, this in black is a melt curve for target A. And you can see that we have actually simulated what stabilization events might look like for that particular target. That's quite easy to do in Prism because you can just fit a Boltzmann curve to your target and you can right shift that by any number of degrees. And the noise around those predicted stabilization events is quite easy to predict, because in our assays, at least, we seem to see that noise and signal are proportional. Having generated those potential stabilization events that we might see, we have a sense of where the assay window might sit at different temperatures, and what a sort of theoretical minimum and theoretical maximum of the assay might look like, what very small stabilization events might look like. We're particularly interested in these sort of 1 to 2 degree stabilizers. We are screening a library of naive chemistry against a target that often doesn't have a known binding site. We're not expecting to find nanomolar binders. It'd be great if we did, but we rarely find nanomolar binders. So we really want to make sure that we're sensitive to detecting those really weak interactions. Here on the left-hand side, you can see that if you plot out those on the y-axis, we've got something called predicted Z prime. This is just an assay statistic. It's a measure of how big your assay window is, how robust your assay is. We like it to be over 0.5. The bigger it is, the better. In general terms, this plot allows us to actually pick or define the temperature at which we should run the experiment. So here, somewhere between 51 and 53 degrees, that's like you would get the best assay window and the best sensitivity to picking up those really small stabilization events. And the other thing that this plot can tell us is actually where we have instances where, for instance, like here, for target B, we do not have a robust assay, and we can only pick up these sort of really large stabilization events. That could be for a couple of different reasons. It could be that the signal is too low relative to the noise, perhaps because your target's expressed at a really low level in your cells. It could be because the slope on your melt curve is really shallow, and so actually it takes a big stabilization event to have a significant effect on increasing the signal. And it tells us where actually we need to do more work on the assay before investing the resource to run a high throughput screen. So this has been a really useful method for us. In terms of how we automate these cell-based thermal shift assays, we would not want to do this by hand. There's lots of steps here, and actually there's more steps even than I've shown you along the top, because along the way you have to CM and DCO your plate. And we ideally want to run approximately about 100 plates a day. This would take about 5 people to do. We're very lucky in high throughput screening. We have these incredible automation platforms. They are one of the highlights and lowlights of working in high throughput screening. If anyone's ever worked with robotics or instrumentation, they have these central hexagonal platforms here with robot arms mounted on the top. And then there's 6 slots for these dockable carts here to be docked on around the hexagon. And each of these carts have their own robot arm just here. And they are loaded up with bits of equipment that we use to run our experiments. So we work with plate-based assays, 384-well plates, 1536-well plates, and we move those plates around these platforms in order to run our experiments. So the Setzer assay starts up here with the plate. We pre-dose compounds into the plate. We have an entire compound management team whose job it is to look after the compound collection and find ways to get it into the plates. We then use this multi-drop combi to dispense some cells and co-incubate the cells and the compound, 1 compound in each well, for an hour. Then we do the thermal shift part of the experiment. So this is the heat step. We only have to heat for 2 minutes at our predefined assay temperature in order to melt our protein just the right amount. And then we do a quick spin just to bring down any condensation off the seal. We lyse with Promega's lysis buffer and bring in the large bit and the luciferin reagent. And then we do another quick spin to spin out any of the unfolded protein aggregate, because we don't want any high bit signal coming from our unfolded protein fraction. And we take this supernatant with the folded protein expressing high bit into a fresh plate using this instrument called the Prime from HiRes. And then we take the fresh plate and we read it in a Ferrostar FSX, which is just a luminescence and fluorescence plate reader. So any plate reader that can detect fluorescence luminescence would be suitable. This is just a shout out to our automation team, because they're absolute wizards who make sure that all of this works, and it's fantastic. And using this setup, we can screen 25,000 compounds a day. So that's enabled us to run, I think now, 3 of these high throughput screens using this technology. Our most recent screen, we ran 500,000 compounds at a single concentration at a single temperature in this experiment. We screened at 30 micromolar, which has been subject to debate in the past, because if you screen at too high a concentration, then you see a whole bunch of artifacts, because compounds will weakly interact with your detection mechanism in your assay and interfere. And in high throughput screening, we're all about trying to find true positives and progress them, and we want to leave the false positives behind. And if you bring too many false positives with you, then the deconvolution gets far too difficult. So we screen at 30 micromolar. If you go too low, then you'll miss some of these weak binders that actually we could be quite interested in as starting points. So for this particular screen, we had 700 actives come out the end, which is a reasonably low active rate. Is kind of what we expected for this particular target, because it's an intractable transcription factor, it didn't have any catalytic activity or known binding site. We then progressed those 700 actives to concentration response, and when you assess these compounds in concentration response, 150 of them gave dose-dependent stabilization, or appeared to give dose-dependent stabilization. We then progressed those compounds to melt curves and assessed whether, when we screen at every single temperature, do we have stabilization? And 72 of those actives were progressed, and they have been progressed to downstream assays in other teams within AstraZeneca. These 72 actives comprised of one chemical series, which is fantastic. That's exactly the kind of answer we want to get out of our high throughput screens, where we're taking lots and lots of compounds and we're cutting down to very small amounts of chemistry. And that whole process takes about 3.5, 4 months. Just a quick comparison of the dye-based DSF that Poppy was talking about and the cell-based thermal shift assays I've been talking about. Dye-based DSF, it needs a lot of protein, right? So if you can make 100 megs of protein, that's great, you can run a DSF. The dye is commercially available, very easy to set up the assay, very quick to set up the assay, and you can get going really, really quickly. On the other hand, if you run a cell-based thermal shift assay, then you don't need any purified protein. Instead, you need your target in a cell line in a way that you can detect it. So you might need antibodies, great if you can get them. If not, you've got to spend time engineering a cell line. And we have a team that does that, and they're pretty quick at it, but it still sucks up time in your drug discovery cascade, right? In terms of equipment requirements, both of these are relatively slim on equipment requirements in terms of you being able to run the assays by hand. We have all the automation. You don't need all the automation if you've just got one target and one compound. You need a qPCR machine to do your DSF, or for CETSA, you don't even need that. You need a machine that is capable of heating at different temperatures and of an endpoint luminescence or fluorescence reader. In terms of scalability, these assays for us are quite expensive. We like to run at sort of 2 to 3 pence per well. And they screen relatively few compounds. Our full collection sits at nearly 2 million compounds. And yet they take about the same amount of time to deliver primary screens as it would for us to screen the whole deck in a different assay technology. The reason that we tolerate that and that we run these screens is because they allow us to prosecute targets that we couldn't otherwise prosecute. So from that point of view, they're fantastic. Just a final note that DSF, you've got a protein-based assay, right? So it's in theory less susceptible to artifacts because it's just one protein in some buffer, and you've got no permeability barriers to getting your compound to bind. On the other hand, cell-based assays have the context of a cell. That's great because you have binding partners and you have cellular localization of your target of interest. That can make it more complex to interrogate and deconvolute your data. And CETSA is really useful if you've got a difficult to express or difficult to fold protein. Quick acknowledgement slide. Loads and loads of people over the years in high throughput screening and cell assay dev have enabled this work. And I think you probably realize as I've been talking that the team goes wider than that. So we have a cell engineering team and a compound management team, and this is quite a big operation. This is just hit discovery, of which we comprise about half, having fun. So yeah, thank you for the opportunity to talk, and any questions for me or Poppy, we'll try to answer.
Thank you for the talk, it's very interesting. I was wondering, how do you analyze the data, especially the raw data? Because if you're generating hundreds of thousands of thermal shift assay curve, for example, to start with, so how do you do that?
Yeah, data management is a huge piece of what we do. So we use a piece of software called Gene Data Screener. Gene Data have a whole suite of different software that can help you analyze your actives. So we literally upload our raw data into their web-based software. And then we have lots of control over how we want to normalize that data, what controls we might want to normalize to, whether we want to mask any wells, whether we have any plate patterns that we want to account for, that sort of thing. We can compare run-to-run data. We can bring in, if you've got a wild type and a mutant, you can compare your curves, that sort of thing. So it's hugely flexible workflow, depending on what kind of assay you're running and what kind of screening you're doing. It can even handle your DSF data with the real temperature curves, right?
Yeah.
And then beyond that, it becomes about data storage and how we report that data to chemists such that they can access it on a database that is permanent.
Thank you so much. I was just wondering what happens when you deal with like multi-protein complexes, or if that's something you ever do tackle, and then how it comes about analyzing that in comparison. Because obviously you mentioned the 2 melt points just from a single protein. So obviously if you have multiple, then you've got multiple factors there.
Yeah, that's a really good question. We've never done it, as far as I know. We've never looked at multiple proteins within the same sample. I guess if you know what your TM shifts or your TM values are for those 2 proteins, could be a way to screen in 2 proteins the same amount of time against the same compounds, and looking at, you know, which TM value is shifted in response to your ligands. So that could be something that's really interesting, but we've never applied it ourselves, as far as I know.
Yeah, in the protein-based world. It's something that's actually quite interesting in the cell thermal shift assays, because we're quite blind to it. So we've tagged one protein of interest, but in theory, if you had a protein that bound tightly to another protein and you bound a ligand to protein B, you could stabilize protein A.
Yeah.
And it's something that we know potentially we have to deconvolute for later down the cascade to check that we've got true target engagement, rather than engaging with a partner of our protein of interest.
Yeah.
Hello. Yeah. In the standard thermal shift assay, part of the signal is reflecting aggregation of the system of the protein, and most proteins aggregate at high temperature. So presumably, if you have a compound that isn't binding at all to the protein, but is reducing that aggregation, that would tend to shift your first derivative, your inflection point, to a higher temperature. So do you ever see cases like that then come back, and I presume these are compounds that are eliminated because you might not see dose response or something, or is this something that you see?
That's a really good question. Yeah.
I've got a sort of an answer for it. If you
Go ahead, please.
you have a library now of frequent hitters, essentially, right?
Yeah.
So that sort of compound you might expect to be interacting in a non-specific manner. So then you might pick it up by looking at your frequent hitters in your DSF technology. But this is why we run our downstream assays, because you know, in NSPR it might look super stoichiometric or something like that. So.
Yeah. And I guess those sort of things you might uncover as you progress the compound through the cascade, and you follow it up in other assays and other technologies and see what it does in those sorts of systems.
There are activities across a range of assays. So we have this it's a binomial survivor function score, so it's called a log BSF score. And if they're active in lots of different assays, they get a very high negative score. So we can look at that. We've also got compounds that are known aggregators in our unwanted mechanism of inhibition set. So we can look at similar compounds in that respect as well. So we've got a range of different spurious activities from different types of mechanisms that we can look at post the screening to see whether, you know, those compounds flag up into any of those sorts of behaviors.
Thank you. I'm still on to data analysis, but in a general scale this time, do you have a way to, how do you correlate the initial phase of your experiments and then the at the end of the line, the good leads for, you know?
Sorry, correlate the what?
Yeah, sorry, I'm not clear. So correlate what you're doing with, you know, with further down the process with the leads that are very useful at the end of the line and for your, you know, for drug discovery.
So I think you're asking, do we follow up and check how successful we are? Is that what?
You could see it like this, but it's not really what I mean in the background. I'm just saying, I'm just wondering how this is done, basically. You know,
how we get from point A to point B.
Yeah, and how do you see the correlation between?
Well, I guess we have the central data. For data management, we have D360, a central database, and you can kind of bring up a selected compound and bring up all the assays that compound's been tested in, up from the point where you do the initial screening to the very end of the cascade, and it's confirmed, you know, in a higher fidelity method. So you can kind of see.
So after we pass on our chemical series to the projects, we then go into what we call a DMTA cycle, so design, make, test, analyze, and the project tries to design chemistry that they think will bind with increasing potency to your target of interest. And you go through several cycles to try and evolve better potency and better chemical properties as well. So if you're looking for particular permeability or lipophilicity, etc. And then you either get there or you don't. And yeah, there is a drop off. Some projects won't succeed and some will.
Yeah. In terms of looking back at things that have come out of, for example, DSF and confirmed in other assays, we do tend to do that. You know, downstream assays, what did they look like in the original DSF? And it's one of the reasons I mentioned that the TM shift doesn't necessarily correlate with affinity, because we've seen ligands that have gone on to confirm that have wildly different TM shifts, very small shifts, very large shifts. So for us, just setting an activity cut where we believe the shift is significant is the best way of going about that and taking everything that's above that cut forwards for downstream testing.
Yeah, I mean, we're in a nice place now with these assays that we are starting to see some of those projects progress to later stages. And actually we can see projects where we know that we reported good equity and projects where we know now that there are maybe artifactual pitfalls that you want to avoid. And so we can learn a bit more about how to set these screens up.
And I just have one last question. In your SETSA, then how good do you have to have the express protein for your detection? Is there?
So we don't like to overexpress our proteins, so we build endogenous cell lines, and that seems to be good enough for the majority of targets that have come in that we've handled so far. So yeah.
Okay, so you don't have to go for have to go for complete massive.
No.
Just enough.
Yeah.
Okay, magic. Any final? Okay, brilliant. Okay.
Well, thank you.